Thank you, Rich. Good afternoon, good morning, good evening, everybody, depending on where you're dialing in from. So the subject of today's webinar is a mine of phenyl boronate affinity chromatography, and this is a technique that's been known and used for many years. It's not really, whilst it's a very useful technique, it's certainly proved its value in a number of applications. It's still not a very widely known technique, there are some quite interesting features of this method that we believe are very useful and are very relevant to biomanufacturing today. So we wanted to use this as an opportunity to share some of the features of this technique of bioseparation and some applications of where this has been used to very good effect. Before we start, I just wanted to briefly give a very short update on Astrea Bioseparations. We were formerly Prometic Bioseparations, and the company has been established for more than 30 years now. We produce an extensive range of bioseparation products, including adsorbents and chromatography columns and pre-packed columns. We're probably best known for our work on small molecule affinity ligands, and we do offer custom ligand developments, custom adsorbent manufacturing services. And I think some of the other webinars in this series speak to some of those services. We have quite significant manufacturing capabilities. We can now produce single batches of affinity adsorbent up to 1,000 litres in batch size. We have quite a significant manufacturing capacity. The main focus, the main use of these adsorbents that we produce is in GMP manufacturing. So we have adopted GMP production standards ourselves, and we offer very comprehensive regulatory support in relation to all of the adsorbents that we manufacture. And to date, over 18 of the adsorbents that we've developed are used in FDA or EMEA licensed products and processes, and we'll speak about some of these later in the presentation. The applications of these adsorbents can be really quite large indeed. We have clients that use our products in columns up to 2 meters in diameter, and we now offer semi-disposable and pre-packed GMP-ready process columns, and we'll say a little bit more about those later in the presentation as well. So today, the company is located in a number of centres between the UK and North America. So our R&D group is based in Cambridge in the UK. Our main manufacturing site is on the Isle of Man. This is our GMP standard site where we have large-scale production of adsorbents. And then in the US, in Canada, we have our manufacturing facility there in Joliet, where we produce our Purabead base beads. And recently, by way of acquisition, we have Astrea Bioseparations US Inc., formerly known as ELS, based in just outside of Boston in Massachusetts. And very recently, we have Nanoparel, who are based in South Dakota. And one feature of the company is that our mantra is discover, develop, deliver. And we are today the only bioseparations company that's able to offer ligand discovery services, adsorbent development services, in combination with large-scale adsorbent manufacture and manufacture and supply of pre-packed chromatography columns to GMP standards. So we have a very broad range of offerings today that we provide to the biomanufacturing community. So now I just wanted to focus on the affinity side and of course boronate. And you know, there's often some confusion about what an affinity ligand is. Is it an antibody? Is it a peptide? Is it an enzyme substrate or inhibitor? In reality, it can be any compound that has selective binding to a target and reversible binding. And that material also needs to be very robust. If it's going to be used in biomanufacturing, it needs to be able to withstand multiple cleaning and use cycles. It should be safe, scalable, and affordable. And of course, the challenge is to find ligands that achieve all of these criteria. And there are many different types of compounds that have been used as affinity ligands over the years, including natural binding ligands such as substrates or inhibitors, antibodies, engineered proteins, peptides, and small molecule synthetic ligands. And the focus of today, the boronate ligand, is an example of this latter category. It's a small molecule synthetic ligand. So the aminophenol boronate ligand is deceptively simple, but as we'll see in a moment, in reality, the mechanisms are actually relatively complex. It provides a very useful tool for the capture and enrichment of target biomolecules that contain cis diols. So these are 1,2 diols in a planar arrangement. And the sort of groups, the sort of entities that have this cis diol arrangement include certain sugar groups, so carbohydrates, glycoproteins, enzymes, RNA, nucleotides, nucleic acids. And so the boronate ligand is very useful either as a capture step to capture and purify these sorts of biomolecules, or to remove them if they're present as impurities. So we'll learn more about this as we go through the presentation. So the mode of interaction of aminophenol boronate. So key to this interaction is the formation of a pair of reversible covalent bonds to form a boronate ester with a target molecule that has a diol. And in the first graphic on the top right there, you can see that the boronate ligand itself exists in an equilibrium in water. And at acidic conditions, it exists as this trigonal planar form, but under basic conditions, it becomes hydrated and it exists as this tetrahedral form, which is a boronate anion. So this is charged. The binding occurs with either the trigonal form or the tetrahedral form. But the tetrahedral form, when it forms these boronate esters, they are much more stable, whereas they're very sort of transient in nature for the planar form. So binding tends to occur at alkaline pH, which is the pH where this trigonal form exists. And the binding occurs, as we mentioned, through the formation of these ester bonds with hydroxyls on the target biomolecule. So in this example here, we've got adenine, which obviously contains with a ribose group attached to it. And that ribose group is an example of a sugar that contains a cis diol. And as we can see here, the tetrahedral form, so this basic boronate ion, is then able to combine with this diol group to form this boronate ester complex. Now this is a reversible complex, it's not a permanent covalent bond. And the binding interaction here is, as we've mentioned, is stronger for this base form than the acid form. And a consequence of that is that by controlling the pH, we can control the binding and elution of the target biomolecule. So just going into this in a little more detail here. So depending on the pH of the environment, our boronate ligand can exist either as the boronate trigonal form on the left or the tetrahedral ion form on the right. This tetrahedral form can readily form these boronate esters, which will then essentially bind them. So with our ligand attached to a chromatography matrix here to immobilized, then it's able to bind a diol entity from solution, and it forms this boronate ester complex. If we then reduce the pH, we can then reverse this equilibrium basically, and also reverse the form of the ligand. So we then revert back to the original planar form of the boronate ion. The binding strength here is very much determined not just by the pH, but also by the nature of the diol. Cis 1,2 diols give the tightest, the strongest binding, followed by 1,3 diols, and then there is a very weak interaction with trans 1,2 diols, but certainly under the majority of conditions, effectively trans diols really don't have any meaningful interaction here. So it's only really sugar groups that contain either cis 1,2 diols or 1,3 diols that have a meaningful binding interaction with the boronate ligand. So why is this this interaction relevant? Well, of course, many proteins contain carbohydrate groups. Glycoproteins, many proteins contain carbohydrate side chains, and these chains are introduced usually at the sort of as post-translational modifications inside the cell. And there's a well-known consensus sequence of asparagine followed by a variable amino acid, and then either serine or threonine. And in the case of an asparagine, it can form N-glycosylated forms. So there's N-glycosylation onto the asparagine groups here. And in the case of serine or threonine, there's O-glycosylation. So this is the way that carbohydrate chains are the way by which they're attached to the polypeptide backbone of a protein. So in addition to glycosylation, there's also a phenomenon known as glycation, and this is well known in plasma, in human plasma and animal plasmas, where after prolonged exposure to sugar groups, the sugar groups become attached to the protein. And in that case, the protein is referred to as a glycated protein. And this is very relevant as it forms the basis of a well-used way of measuring diabetes by measuring the amount of glycated hemoglobin in relation to the non-glycated hemoglobin. And we'll see an example of this later. So the sorts of sugar groups that we're finding in glycoproteins that contain diols, there's quite a range of these. Galactose, mannose, sialic acid in particular, mannose and sialic acid form very strong complexes with amino phenol boronate. You can see here there are some 1,3 diols such as xylose. And in the case of nucleic acids and nucleotides, then they often contain ribose, and ribose also contains a 1,2 cis diol. So these are the sugar residues that are able to form complexes with amino phenol boronate. And the majority of these, certainly the pyranose, the 6-membered ring forms, these are commonly found in the majority of glycoproteins. So amino phenol boronate is a very good way of separating glycoproteins from non-glycoproteins. So there are some other features of these interactions that we need to take into consideration here, because the ligand itself has a hydrophobic core structure, the phenyl group. And of course, hydrophobic groups can participate in hydrophobic interactions. And also the tetrahedral form of the ligand here, the boronate anion, is negatively charged. So that means in addition to the specific binding interaction with diols, there's also the potential to form either hydrophobic interactions or ionic interactions, electrostatic interactions, in a more non-specific way. And on occasion, it's necessary to reduce these non-specific binding interactions. And in the case of ionic interactions, the main way of doing this is by adjustment of the ionic strength of the solution. So this can be achieved by the addition of salt, of sodium chloride, or increasing the concentration of the buffer. It's important not to increase the buffer concentration too much, because if we go to very high ionic strengths, they will promote hydrophobic interactions, and we don't want to do that. So we have to keep the ionic strength at a sort of low to moderate ionic strength. Also, there's a potential here with certain proteins that, as well as the attraction of by these non-specific electrostatic interactions, there can also be repulsion. If the protein itself is negatively charged, it can be pushed away from the ligand. So again, adjustment of the pH is often important here to modulate these unwanted interactions with the ligand. In a recent study, an investigation into the types of salt that can be used to modulate the binding, it was found that sodium fluoride or ammonium sulfate are particularly useful for removing these undesirable electrostatic interactions while maintaining the desired ester formation with the boronate group. And this work was undertaken with a chromatic boronate adsorbent, using monoclonal antibodies as an example. Monoclonal antibodies, majority of antibodies are glycosylated. So the use of aminophenol boronate is a very good way of isolating glycosylated antibodies. And again, we'll see an example of that shortly. Mannose and sialic acid are particularly effective in binding to boronate groups. And because of that, proteins that have predominantly contain either mannose or sialic acid, we can use a lower pH with these proteins. And that reduces also any electrostatic interactions then with the ligand, and prevents the binding of non-monosylated and non-sialylated glycoproteins. So other glycoproteins that contain different sugars will not bind at lower pHs, whereas the mannose or sialic acid containing glycoproteins will bind. So once we've bound the ligand, how do we elute? Well, as we've already mentioned, we can use acetic acid. This is, acetate buffer, typically at pH 4. It's a very effective way of reversing this equilibrium and releasing the diol containing compound. There is another way of achieving elution, which is also very effective, and that is to add a competing diol. Often sorbitol is used. And sorbitol has an abundance of diols here, and it forms very tightly bound ester complexes. And because binding is an equilibrium, it means that it's able to displace less tightly bound diols from the boronate ligand. So in the case of the lowering the pH, we're actually removing the boronate ion that participates in binding, and in the case of sorbitol, we're adding a competing diol which displaces the bound material. And after use of sorbitol, we then have to wash the adsorbent with a low pH buffer. We have to condition the adsorbent to remove the bound sorbitol before we can then reuse the adsorbent for the next binding cycle. So applications of boronates in bioseparations are quite varied. So we can use them to capture and purify glycoproteins that contain these diol-containing sugar groups. We can use them to remove glycosylated or glycated impurities to separate these away from proteins that don't have carbohydrate groups or don't have sugars that contain cis diols. We can also use this. We can use this binding effect in the case of glycated proteins to actually quantify the amount of glycated protein relative to non-glycated protein. And in the case of hemoglobin, this forms the basis of the very well-known HB1AC test, which is the standard classic test for diabetes, because depending upon the glucose level in blood, the higher the glucose level over a long period of time gives rise to a higher level of glycated hemoglobin. And this is almost a linear relationship. So the higher the level of the HbA1c form of hemoglobin, the greater the sugar content of the blood over a long period of time, and essentially the more diabetic that patient is. We can also use this as a means of separating DNA from RNA. And of course, DNA, both DNA and RNA have ribose groups, but in the case of DNA, it's a deoxyribose group, so it's missing a hydroxyl, whereas ribose has both hydroxyls that form this cis diol. So DNA does not bind to boronate, whereas RNA does. So it's a very effective way of resolving and purifying RNA from DNA. Boronate also has some other interactions with enzymes, particularly enzymes that have pockets, such as serine proteases, and it can interact at the active site, almost as an active site binding inhibitor. And I'll show some examples where this ability of boronates to bind to certain enzymes has been used to very good effect. It also binds things like cofactors, for example, such as UTP or NADP that also contain ribose. We can use it, as I've mentioned, as a way of capturing and separating glycosylated monoclonal antibodies, and we can use it to remove things like RNA away from CC plasmid DNA. So there's quite varied uses of boronate ligands. So the aminophenol boronate P6SL product that Astrea produces, we've manufactured this for a number of years. It's one of our larger volume products that we manufacture. It's attached, the ligand is attached to Purabed, the P6XL version of Purabed. The bead size is around 100 microns, and this material is able to bind up to around 30 mg of sorbitol per mil of adsorbent. It can support very high flow rates, so operational flow rates certainly up to 500 centimeters an hour, and it can also support fairly high pressures, certainly up to 3 bars or 45 psi. It has a very wide operating pH range, so it is usable in the pH range 2 to 14. For long-term storage, we recommend this maintained within the range of sort of pH 3 to 12 or 3 to 13, but it is compatible with molar sodium hydroxide for shorter durations, such as column cleaning. So it's a very stable, a very relatively robust adsorbent, which are very desirable properties for use in manufacture. And because of that, and because it can be cleaned very effectively with sodium hydroxide, it can be used for many cycles. So just a little bit about Purabed. This is our neomonodisperse agarose beads that Astrea has manufactured for a number of years now, over 20 years. The process we use to produce these beads is very different from the traditional ways of making porous agarose beads. So we use a spray atomization process. So this is a patented method that is owned by Astrea, that gives rise to a very uniform particle size. And that has a number of advantages, benefits in use. So it means that because the beads are all the same size, the flow channels between the beads are also the same size. So these beads have really excellent flow properties. They're relatively easy to use. They run at relatively low back pressures. They will support high pressures, but they will certainly run at relatively low back pressures. The beads have a very high surface area. They're highly porous, and they're very robust, long life materials. And of course, we're quite proud of the fact that we've developed this spray atomization process, because one of the benefits of this is it does not use large volumes of organic solvents like the historical processes do. So this is a low carbon green production process now for agarose beads. In terms of the pressure flow relationship, you can see that here. So this adsorbent is quite capable of supporting flow rates up to 1,000 centimeters an hour at pressures of around well, less than 2 bar at that. So it's very pressure resistant, and it can support very high flow rates. And in terms of its pH stability, as we mentioned, it's very stable, and it's stable intermittently at pH 14, which is certainly for several days continuous exposure. So this means it can be effectively cleaned and sanitized in use. The recommended conditions for purification, we recommend generally a low to medium ionic strength. You recall we mentioned just earlier about the importance of controlling the ionic strength and not having it too high or too low in some instances. Generally speaking, the pH should be alkaline to promote the formation of the boronate ion. So that means the pH for binding generally is in the region of pH 8 to 9.5, and usually between pH 8.5 and 9. A whole variety of buffers can be used here. Phosphate and glycine work particularly well. Flow rates for binding, we recommend, in the range of 100, 150 centimeters an hour for binding. It's important it isn't too high to give the glycosylated protein the opportunity to diffuse into the beads and diffuse out again. And as we mentioned, elution can be achieved either using a low pH buffer such as acetate, or the addition of a competing diol such as sorbitol. But again, if sorbitol is used, it's really important that it's treated with a low pH buffer before it's then used again for the next binding cycle. So we'll look at some examples now, and one protein fairly well known for use in enzyme immunosorbent assays is alkaline phosphatase. An alkaline phosphatase is a glycoprotein. It contains fucose and N-acetyl glucosamine groups. So this is a relatively small scale purification here. We have alkaline phosphatase in buffer at pH 9. We then load this onto our pack column, wash with the same buffer, and then we elute here with 200 millimolar sorbitol at pH 9, and then followed by a 1 molar sodium hydroxide cleaning step. And as you can see here, the alkaline phosphatase is quite effectively bound, and we have a very sharp elution peak when we add sorbitol. And again, sorbitol is now competing for the formation of this boronate ester. And you can see from the SDS page gel there that this isn't a pure alkaline phosphatase. There are other proteins present. The majority of those other proteins that flow straight through, they're not bound, they don't have diols. We see some breakthrough of the alkaline phosphatase, but that may be due to the fact that often with glycoproteins, we see different glycoforms of the protein. And certainly if there's been any cleavage of the carbohydrate group, then it will not bind to the boronate ligand. So one advantage of aminophenylboronate chromatography is that it's a good way of enriching the glycosylated forms of the same protein, of removing the non-glycosylated form of that same protein. So sort of target protein impurities can be removed here using this boronate technique. The eluted enzyme is relatively pure, as you can see here, and it's recovered in fairly high yield. So this is a very effective way of capturing and purifying alkaline phosphatase. As we've mentioned, IgG also is also glycosylated, has carbohydrate groups. In particular, mannose. It often contains mannose and groups and N-acetyl glucosamine as well. Generally speaking, glycosylated antibodies are able to bind very effectively to the boronate ligand. So in this example, we're using plasma-derived polyclonal IgG. And of course, being plasma-derived, it contains the carbohydrate groups that are quite variable. They're variable in length, they're variable in content. So not all of these will bind with the same strength to the boronate ligand, depending upon the number and type of the sugar groups attached. So again, this is a relatively small scale purification here. We're adding the polyclonal IgG. It's relatively pure to the column. We're eluting again with sorbitol and cleaning the column with sodium hydroxide. And as you can see here from the SDS page gel, that we see very efficient binding of the IgG. All of the IgG is bound. And the IgG then is then eluted, and you can see here we see, although it's an isocratic elution regime, we see 3 peaks coming off. Now these are almost certainly different glycoforms of IgG. This is reflective of the fact that these are different types of sugar groups, different numbers attached to these IgG molecules, and they're binding with greater or lesser affinity to the ligand, by the formation of these boronate esters. So that's the reason for this peak. So we can use this as an effective way of separating out the different glycoforms of polyclonal IgG, or in the case of a monoclonal IgG, it will be a very effective way of capturing and purifying the particular glycoform of interest. Another way of using the adsorbent is to remove glycoprotein impurities. Now, a number of recombinant proteins are produced in yeast expression systems, and yeast is well known for hypermanosylating proteins. It tends to add mannose onto proteins. Certainly the yeast host cell proteins are often quite rich in mannose. And as a consequence, these monosylated proteins can be removed using amino phenyl boronate chromatography. And it's important in many cases that they are removed, because some of these yeast glycoprotein impurities are antigenic. These form the basis of yeast antigens, and a small proportion of the population are actually quite sensitive to yeast antigens. So it's important that these are removed from therapeutic products produced in yeast. So again, in this example here, we're binding at pH 9. We're eluting in this case by the use of an acidic buffer, and cleaning with sodium hydroxide. And as you can see here, this is a very effective way of separating the yeast glycoproteins from the non-glycosylated protein. The recombinant protein that's been expressed in the yeast here is not glycosylated, so it passes straight through the column unretarded. Whereas the yeast glycoprotein impurities are bound, and you can see that on the right there on the Western blot, and you can see a very substantial removal of these yeast glycoproteins. This is a glycoprotein stain that's been used here on the Western blot. So this is a very effective way of removing glycoprotein impurities from a non-glycosylated target protein. We can also use this, as I've mentioned, with glycated proteins, and this is the classical separation of glycated hemoglobin from non-glycated hemoglobin. And you can see here that the non-glycated hemoglobin, so this is a red cell lysate. We lyse the cells just in water, and that will release the hemoglobin, and that the non-glycated hemoglobin will pass straight through the boronate column unretarded. But the glycated form will bind, and it's only removed when we elute it with the elution step here, and by lowering the pH. And as you can see, we can see quite a clearly defined elution peak here. And the size of that peak is directly related to the concentration, to the relative proportion of glycated to non-glycated hemoglobin. So this is a really excellent way of resolving these two components, and it's the reason why it's so widely used as the primary diagnostic form for diabetes, much more so than a blood sugar test, because this shows long-term exposure to high levels of glucose in the blood. And then the final example here, this is an enzyme. I mentioned earlier that certain serine proteases can also bind to the boronate ligand. It acts as a selective binding ligand to the active site of the enzyme. This is an example. It's actually a marine serine protease that's very effectively bound and purified. And this is used. This is one of several commercial applications of the boronate resin. In another case, it also binds to enzymes such as thrombin. So the boronate adsorbent is used commercially for the manufacture of thrombin, which has a number of clinical applications. So in terms of the availability of these materials, for availability for research purposes, we have a number of formats here. We provide small 96 well micro column screening kits, and we can certainly provide these kits with the boronate adsorbent for rapid sample processing or for optimization of binding and elution conditions. We can supply larger 1 mil and 5 mil HT columns, and these attach directly to actor systems, for example. And then we also have larger Evolve R columns, which are 10 and 50 mil volume. These are pre-packed columns, which again can be attached directly to an actor system. For larger volumes, we have a whole variety of adsorbent pack sizes we offer. So for laboratory use, we have pack sizes in the range of 25 mils to 1 liter. For process applications, we produce packs in 5 and 10 liter sizes, and we can produce batches of this adsorbent up to 1,000 liters, and we regularly produce batches around 250 liters in volume. And we can also provide these as pre-packed columns with our Evolve D columns. So just to finish now on the Evolve D column range, we have it's relatively new. We introduced these, we launched these last year. So these are GMP standard pre-packed columns. We have a range of column diameters, currently 7, 10, and 20. We're in the process of developing larger diameter products at the moment. And we offer different bed lengths as well, 10 and 20 centimeter bed lengths. They operate at quite significant flow rates. They're very robust as packed columns. All of the components conform to the FDA and USP requirements in terms of leachables and extractables. And these are fully compatible with use in GMP processes. So we're able to provide all of our adsorbents, including the boronate, the aminophenol boronate adsorbent, in a ready-to-use pre-packed column format. So at that point, we'll finish the presentation and be very happy to take any questions that you have. This presentation will be available later, and you can see here the contact details if you would like to contact us in relation to this presentation, the aminophenol boronate adsorbent, or any other purification problems that you would like to discuss with us. So thank you for your attendance today.